Good morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here.
This is Andrew and Renee. Welcome to Stack Series.
Today, we are exploring the revolution in drug discovery.
And this is a topic with some serious weight to it.
It really is. I mean, we often cover technology or business, and those are fascinating, but this is different. This is about life and death. It's the difference between a diagnosis being, a manageable condition or a terminal sentence.
It's the engine room of modern medicine, really. But the thing is, for the longest time, that engine has been, let's just say, incredibly inefficient.
That's a very polite way of putting it.
Well.
Historically, finding a new drug, it's always been described as looking for a needle in a haystack. But as I was going through the material for today, I realized that analogy it's actually way too optimistic.
It is, yeah, because it implies, what a needle looks like.
Right, and that it's definitely in there somewhere.
Exactly. A better analogy would be something like, you're looking for a needle in a haystack, but the haystack costs $2 billion to build.
Okay.
It takes you 15 years to search through it. And then half the time, when you finally pull out something sharp, it turns out to be a thorn that actually hurts the patient.
Instead of a needle that cures them. That's the reality we've been in. High failure rates, just astronomical costs.
Yeah.
But this is where it gets really interesting. We're recording this in early 2026. And if you look at the landscape right now, the tech, the papers being published, it doesn't just feel like we're getting better at searching the haystack.
No.
It feels like we're about to burn the haystack down and just use a giant magnet.
That's a great way to put it. We are absolutely at an inflection point. And I don't use that phrase lightly.
It gets thrown around a lot.
It does. But for the last century, biology has basically been a descriptive science. We were just observers. We, looked through microscopes, we drew pictures, we described what we saw. But now, as we're moving through 2025 and into 2026, biology is fundamentally transforming. It's becoming an information science.
So it's all about data.
It is entirely about data. It's not just about pipettes and petri dishes and, people in white coats holding up test tubes anymore.
Right.
It's about this convergence of massive data sets, artificial intelligence, atomic level imaging, and even the dawn of quantum computing. It's becoming an engineering discipline.
So our mission today is to really unpack that revolution. We're going to look at how we moved from what scientists used to jokingly call blobology.
Blobology, yeah, I love that term.
These blurry images of proteins to actually seeing individual atoms. And we're going to talk about how computers are now designing drugs completely from scratch.
Instead of us just hoping we stumble across them.
Exactly. It's like they're hallucinating cures into existence.
We'll be breaking this all down by looking at what some are calling the top 5 drug discovery highlights of 2025 and looking ahead to what 2026 holds. It's a lot of ground to cover. We're talking the 10 V's of big data, cryo-EM, AlphaFold 3.
And quantum mechanics. So let's get into it. I think we have to start with the foundation. We hear the term big data thrown around constantly.
All the time.
It's in every tech article, every pitch deck. It kind of feels like a buzzword that's lost its meaning. But in this context in pharma, it seems like it's the actual bedrock of this entire shift.
It is the bedrock. You absolutely cannot have the AI revolution without having the data revolution first. But it's really crucial to understand that big data in drug discovery is a completely different beast than just having a really, really big Excel spreadsheet.
Right. So what does it actually mean here? Because I think for most people, it's just, a full hard drive somewhere in the cloud.
It is that, but the complexity is where the real devil is. In some of the source material, they break it down using this framework, the 10 V's of big data.
To 10 V's?
Yeah, it's a way to get your head around why this data is so remarkably hard to wrangle. The first two are the ones everyone kind of knows, volume and velocity.
Volume is just the size, the sheer size of it.
Exactly. But let's try to contextualize that size for a second. Take PubChem, which is one of these massive public repositories for chemical compounds.
Okay.
As of the numbers we have, it holds over 102.4 million compounds and 1.1 million bioassays.
Wow.
We're talking terabytes and terabytes of data just for the structures alone.
And that's not even the results of the experiments. That's just the library of stuff. That's the inventory.
It's just the inventory, exactly. Then you have the second V velocity. This is the speed at which new data is just hammering the servers.
Right.
In the old days, a lab might test, 10 compounds a week?
If they were lucky.
Right. Now we have high-throughput screening robots, literal automated arms, testing thousands of compounds a day.
And you've got patient data coming in from wearables, real-time clinical trial updates.
The data pipe is a fire hose, and the valve is just stuck wide open.
Okay, so we've got a ton of it, and it's coming at us really fast.
Yeah.
That still sounds manageable if you just have enough computers.
Yeah.
But I feel like the third V is where the real headache starts for the scientists.
Oh, yeah.
Variety.
Variety is the nightmare. I would say this is the single biggest barrier to applying AI effectively in medicine.
So why is it so much harder than, say, financial data?
Well, think about it. In banking, your data is all numbers, right? Dollars, dates, percentages. It's structured. It fits neatly in a spreadsheet.
Yeah.
In drug discovery, the analogy you used earlier was a good one. It's like trying to organize a library where some books are written in English.
Some are in binary code.
Some are audiobooks, and some are just loose pages of handwritten notes scribbled on a napkin by a doctor who hasn't slept in 24 hours.
That's a very vivid picture. I can see the problem. You're trying to merge completely different realities.
Exactly. You've got chemical structures, which are geometric shapes, their topology. You have genomic data, which is just sequences of letters A, C, G, T.
The string of text.
Right. Then you have pathology images, which are visual pixel data. And you have clinical trial notes, which is just natural human language. Trying to merge all of these different data types, what we call multimodal data, into something a single computer can understand all at once is. It's incredibly difficult.
It's like trying to get an AI to read a book where page 1 is a diagram, page 2 is a song, and page 3 is an actual biological tissue sample.
That is a perfect analogy. And that flows directly into the next big V, which I found fascinating. Veracity.
Or in plain English, quality.
Quality. Just because you have a mountain of data doesn't mean it's true, right?
Right. The old garbage in, garbage out principle.
Or as I've heard some of the more cynical data scientists put it, trash in, trash out.
Mm-hmm.
And public data is notoriously messy. Different labs run experiments in slightly different ways. Maybe the temperature in the room was off by 1 degree.
Or the reagent they used was from a different brand.
Exactly. There is inherent uncertainty in all of it. So if you feed an AI model data that says drug A kills the cancer, but that experiment was flawed, the AI will just confidently predict a cure that doesn't actually work.
And I noticed the sources also brought up vocabulary. This seems like such a basic problem, but don't we have standard names for things?
You would really hope so, but no, not really. Different labs, even different databases, they all describe the exact same molecule using different terms or formats.
So, what, like one calls it by its trade name, another by its chemical name?
That's part of it, but it's also the format. You might have one database using what's called the SMILES format, which is basically a text string that represents a chemical structure, and another one using something called INCHI keys.
So it's like trying to compare a book by Mark Twain with a book by Samuel Clemens, and the computer has no idea it's the same author.
Precisely. And if the computer thinks they're two different drugs, it misses the pattern. It can't make the connection.
There were a few other V's mentioned. Volatility, validity. But the one that really stood out to me as the bottom line was value.
The ultimate goal. At the end of the day, all of this effort, the cleaning, the sorting, the storing, the translating, it's only worth it if it actually reduces the cost of developing a drug.
Right. If we spend $1 billion on data management and it doesn't make drugs cheaper or faster to get to patients, we've failed.
We're completely failed.
So speaking of where all this data lives, let's talk about the landscape of these databases. It's not just one big folder on a server labeled science, is it?
No, it's highly segmented. You really have 3 main buckets of knowledge that researchers pull from.
Okay.
First, you have what are called chemical collections. These are basically the catalogs. We mentioned the Anamine Real database, which has something like 700 million compounds, or ZINC, which has over 230 million.
So these are just giant shopping lists. Here are all the things that exist or that we think we can make.
That's a great way to put it. Then second, you have the target databases. These are more like the instruction manuals. These are places like BindingDB or Shemblewell. And these are often manually curated, which is a key difference.
Meaning a human scientist has actually looked at the published literature and verified the data point before it goes in.
Exactly. So the quality is generally much higher. These databases tell you, does this specific drug actually stick to this specific protein?
And then the third bucket is the clinical databases like clinicaltrials.gov.
Which has, I think the number was over 324,000 research studies. This is where you find out if the drug actually worked in people, or more often, if it failed.
But here's the catch, and I think this is a really critical distinction for anyone listening.
Yeah.
There's a huge difference between quantity and quality.
Yeah.
The sources pointed out something really telling about PubChem. Just because it has millions and millions of data points doesn't mean they're all hits.
That's a crucial nuance. Let's take the drug acyclovir, for example. It's a well-known antiviral. It's used all the time.
Right.
In the PubChem database, it might have, say, 13 active responses, meaning it did something positive in 13 specific tests. But it also had 204 inactive responses. It failed in 204 other experiments.
And usually, in science, we only publish the success. We kind of hide the failures. But for big data, that failure data is just as important, isn't it?
It is absolutely vital. If you only teach an AI what works, it becomes hopelessly optimistic. It develops a massive confirmation bias.
It has no sense of what's impossible.
Exactly. It needs to know what doesn't work to understand the boundaries of biology. It needs to see the wall to know where the door is. The reality is most experiments fail. That's just science. And the data has to reflect that reality.
So we have this massive, messy, fast-moving ocean of data. We have the shopping lists of chemicals. We have the records of what worked and what didn't. But to cure a disease, you usually need to do more than just read a list.
You need to see the enemy.
You need to see the target. You need to see the lock before you can even think about cutting a key. And this brings us to the second major pillar of our discussion, the visual revolution.
Visualizing the invisible.
For a very long time, the gold standard for this was X-ray crystallography. And I've seen those images. They're beautiful, like abstract art. But that method had some really serious limitations, didn't it?
It did. I mean, don't get me wrong, X-ray crystallography is a brilliant technique. It won Nobel Prizes for a reason. But, as the name implies, it requires you to force a protein to form a crystal.
And what does that mean exactly?
Think of a crystal like a brick wall. Everything is ordered, it's repeating, it's rigid, perfectly aligned.
But biological things, proteins, enzymes, they aren't bricks.
No, they're machines. They're dynamic. They wiggle, they vibrate, they change shape to do their jobs. And many of the most important ones, specifically the membrane proteins that sit in the oily shell of a cell, they absolutely refuse to crystallize.
Why is that?
They're just too floppy or too greasy. It's like trying to stack jelly. So for decades, these critical drug targets were effectively invisible to us.
And to really understand why seeing these structures matter so much, why we can't just guess, I think we have to talk about thalidomide. This story really struck me. I knew about the tragedy, of course, the birth defects in the 50s and 60s.
Oh, terrible, terrible story.
But I didn't realize the sheer scientific mystery behind it for so long.
It's a heartbreaking but absolutely crucial example of why structure is everything. So thalidomide was marketed as this completely safe sedative, a cure for morning sickness. It was a wonder drug. And then it caused these devastating limb malformations in thousands and thousands of babies. It was pulled from the market in disgrace. But here's the thing. For decades, scientists had no idea how it caused those defects.
It was a total cold case.
A complete cold case.
Right.
We knew the weapon thalidomide. We knew the victim, the developing fetus. But we had no idea about the mechanism. Was it attacking DNA? Was it blocking some nutrient? Nobody knew.
And what's so strange is that today it's actually used to treat multiple myeloma, a type of cancer. So the drug itself wasn't evil. It was just profoundly misunderstood.
Correct. But the breakthrough, the solution to this decades-old mystery, it finally came in 2010.
50 years later.
50 years later. Researchers finally managed to crystallize thalidomide while it was bound to a protein called cereblon. And cereblon is part of something called a ubiquitin ligase complex.
Okay, that's a mouthful. What is a ubiquitin ligase?
Think of it as the cell's built-in trash disposal system.
Okay.
Its job is to go around and find old or broken proteins and tag them with a little destruction sticker, a molecule called ubiquitin, so the cell's recycling machinery knows to grind them up.
So it's the janitor of the cell.
It's the janitor. And when they finally got that crystal structure, that picture from 2010, what did they see?
What did they see?
It was like finding a tiny speck of dust jammed in a watch gear. Thalidomide was this tiny molecule nestled in a pocket on this huge protein, but just by being there, it changed the shape of that pocket ever so slightly.
So the trash collector started picking up the wrong trash.
Exactly right. Because the shape of that pocket changed, the ubiquitin ligase, with thalidomide's influence, started grabbing a whole new set of proteins. Specifically, it started grabbing transcription factors that are absolutely essential for limb development in a fetus.
And it tagged them for destruction.
It tagged them for destruction. So the drug didn't attack the limbs directly. It tricked the cell's janitor into throwing away the blueprints for building the limb.
That is an incredible mechanism, and we only understood that because we could finally see the structure.
Only because we saw the molecule sitting in the pocket.
And once they saw that, a huge light bulb went on. They must have realized, wait a minute, if we can trick the janitor into destroying good proteins, can we trick it into destroying bad proteins?
Precisely. This single discovery gave birth to a whole new field of medicine called targeted protein degradation. Can we design new drugs that hijack this trash disposal system to specifically destroy cancer-causing proteins, or the proteins that cause Alzheimer's? This entire field only exists because we could finally see that structure.
But it took X-ray crystallography 50 years to get that one shot of thalidomide. We can't wait another 50 years for the next one. We needed something faster, better, and something that could see all those uncrystallizable proteins.
Which brings us to cryo-electron microscopy, cryo-EM.
This is the game changer?
This is the game changer. The sources call it the resolution revolution, and that is not hyperbole.
So walk us through it. How is it different from the crystal method?
Well, instead of spending months or years trying to grow a perfect crystal, you take your protein in a solution, just in its natural, wiggling, happy state, and you flash freeze it.
Flash freeze.
You plunge it into liquid ethane so incredibly fast that ice crystals don't even have time to form.
Good.
The water just turns into this glass-like state called vitreous ice.
So protein is basically frozen in time, mid-motion, like a bug in amber.
Exactly. And because you have millions of copies of that protein in that single drop of water, all frozen in slightly different poses, you can then take pictures of all of them using a very powerful electron microscope.
The analogy that really helped me get this was the difference between a regiment of soldiers and a dance floor.
Yes, that's the perfect way to think about it. X-ray crystallography is like taking one single, very clear photo of a regiment of soldiers standing perfectly still in formation. You get a super sharp image because they're all aligned.
Right.
Cryo-EM is like taking a million high-speed photos of a crowded dance floor. Everyone is doing something slightly different. Some are facing left, some are facing right, some are mid-spin.
And if you just took a long exposure of a dance floor, you'd just get a blur. A total mess.
A total mess. But if you have a powerful computer that can look at those millions of individual snapshots of the frozen dancers and say, okay, that guy over there is doing the waltz and that person is doing the tango, and oh, look, here are 10,000 people all frozen in this exact same pose.
You can average those 10,000 identical poses together.
And you get a crystal clear 3D reconstruction of that pose. And you can do that for every pose. You can see the whole dance. You catch the proteins in their natural functional states.
Right. And for a long time, this didn't work very well, right?
Either.
This is where the term blobology came from.
It did. The images were just fuzzy blobs. You could see the general shape. Oh, it kind of looks like a donut, but you couldn't see the atoms. You couldn't see the details you need to actually design a drug.
What changed? Why are we talking about this in 2026 as the new gold standard?
It was really a convergence of hardware and software. The Nobel Prize in 2017 went to the pioneers, Dubochet, Frank, and Henderson, who laid the groundwork. But the real acceleration came from the cameras. We moved from using physical film, yes, they used actual film in electron microscopes.
That's wild.
To these new direct electron detectors.
Which, funny enough, is directly related to the technology in all of our cell phones.
It is. The massive consumer demand for better and better smartphone cameras drove sensor technology forward at an incredible pace. These new detectors are unbelievably sensitive. But crucially, they take movies instead of still photos.
Now, why would you need a movie of a frozen protein? It's frozen solid. It shouldn't be moving at all.
You would think so. But when you blast a tiny protein with a high-energy beam of electrons, it actually moves. It jiggles from the impact. And that jiggling creates blur.
Ah, okay.
But if you're taking a movie, a high-speed video, the computer can track that movement frame by frame and computationally correct for it. It's called motion correction. And that's what turns a blurry blob into a sharp, beautiful image where you can actually see the side chains of individual amino acids.
That is incredible. So we have literally gone from blobology to seeing individual atoms. And now we can finally see all those membrane proteins that refuse to crystallize.
Which, by some estimates, represents about 60% of all drug targets. It's like the lights have suddenly been turned on in a room that was dark for 50 years. We can finally see the receptors that control pain, the ion channels that control your heart rhythm, the proteins that viruses use to get into our cells.
But dealing with these movies, I mean, you're talking about terabytes of high-resolution video. That sounds like a data nightmare all over again. We're right back to the volume and velocity problem.
It is a huge data problem. A single cryo-EM session can generate terabytes of raw data in a single day. And processing that used to require a dedicated supercomputer in the basement of the university.
Which not everyone has.
This is where companies like CryoCloud have come in.
I saw that in the notes. This is basically moving all that microscope processing into the cloud.
Exactly. It completely democratizes the science. You don't need a massive multi-million-dollar IT infrastructure in your own lab anymore. You can stream the raw data straight from the microscope to the cloud. It gets processed on these massive clusters of GPUs, the same kind used for high-end gaming and AI, and you get your finished structure back.
And it's secure, which is key for the big pharma companies.
Absolutely critical. It's ISO 27001 certified. Because if you're Pfizer or Merck and you just solve the structure of a billion-dollar drug target, that structure is your intellectual property. You can't just upload it to some random public server.
Okay. So we have the big data foundation. We have the cryo-EM eyes to see the targets in atomic detail. Now we need the brain to make sense of it all.
And this brings us to the third section. Artificial intelligence.
This is where the speed really, really picks up.
This is where we move from just observation to actual prediction.
Now, we've been using computers in drug discovery for a while, haven't we? I remember hearing about CAD, computer-aided drug discovery, way back in the 2000s.
Yes, CAD has been around for decades. But it's important to understand that the old way was fundamentally different from what we're calling AI today. The old way relied on things like QSRR,
which stands for?
Quantitative Structure-Activity Relationship.
Right, another mouthful. What does that actually mean in practice?
In practice, it was basically a very complex spreadsheet correlation. The scientists defined the rules. We said, okay, computer, listen up. If a molecule is greasy, what we call having a high log P, then it might cross a cell membrane. If it has a hydrogen bond donor right here, it might bind to this spot. It was rule-based. We told the computer exactly what to look for based on what we already knew from chemistry.
So the computer was really just checking our homework.
It was just checking our homework, exactly. But the new way deep learning and neural networks is completely different. We don't tell the computer the rules. We just show it the data, all those millions of compounds in PubChem, the failures, the successes, and we say, you figure out the rules. You tell us what matters.
And a great example of this from the research was the deep tox model.
Right. So this came out of the Tox 21 challenge, which was this big competition to see if computers could predict if a chemical would be toxic to humans. The deep tox model used deep learning to look at thousands of chemical structures, and it predicted toxicity better than any other method.
And the crazy thing was,
it found patterns that human scientists hadn't noticed. It saw these subtle correlations in the molecular structure that just screamed danger, which we had been missing for years.
But the headline grabber, I mean, the absolute rock star of AI in biology right now, it has to be AlphaFold. And specifically, what we're seeing now with AlphaFold 3.
Absolutely. AlphaFold just changed the entire game. Google DeepMind basically solved what was a 50-year-old grand challenge in biology, the protein folding problem.
Which is predicting the 3D shape of a protein just from its genetic sequence.
Right.
But I want to push on this a little. AlphaFold 2 came out a couple of years ago, and it was a massive deal. It gave us the shape. So why is AlphaFold 3 such a huge headline now in 2025 and 2026? Is it just a slightly better version?
No, it is a categorical leap forward. You're right, AlphaFold 2 gave us the statue. It showed us what the protein looked like all by itself. But drugs don't work in isolation. Drugs work by touching things.
Right.
AlphaFold 3 predicts the structure of proteins and how they interact with other things, with DNA, with RNA, and most importantly, with small molecules, with drugs.
So it's not just a statue of the protein anymore. It's predicting the handshake between the drug and the protein.
Exactly. It's simulating the actual interaction. It predicts where the drug will bind and how the protein's shape will change and react to that binding. This is what allows us to screen billions of compounds virtually. We can just ask the AI, does this drug shake hands with this cancer protein, without ever having to touch a test tube?
Although I did notice a very important note of caution in the sources. AlphaFold is amazing, but it is still a prediction. It's a very, very educated guess. A hallucination, in a way.
Trust but verify. That is the absolute mantra in the field right now. You still need experimental validation, like cryo-EM, to prove that the AI's prediction is correct in the real world. It's a hybrid world. The AI tells you where to look, but you still need the microscope to confirm that you actually found it.
There was another AI concept in our materials that sounded super sci-fi, and to be honest, a little intimidating. AGIMA.
Ah, yes.
Atomic graphs with intermolecular adjacency.
Yes. This is a really fascinating and powerful new approach to understanding, specifically how drugs bind to their targets. It solves a specific problem with how computers have traditionally seen molecules.
Okay, so break this down for us, because graph neural network sounds like something straight out of a PhD thesis.
It does, but the concept is actually pretty intuitive. Think about a photograph. A photograph is just a grid of pixels. For a long time, we treated molecules like flat pictures. You know, those 2D drawings of hexagons and lines you see in chemistry class?
But molecules aren't flat drawings. They're 3D objects with forces and charges.
Exactly. So AGIMA uses what's called a graph neural network. The best analogy is to think of a social network like Facebook or LinkedIn. You have people and you have friendships connecting them.
Okay, nodes and edges. I can visualize that.
Right. In a molecular graph, the atoms are the people, the nodes, and the chemical bonds between them are the friendships, the edges. But AGIMA does something really clever. It looks at intermolecular adjacency.
Which means?
It means it doesn't just make a graph of the drug and a separate graph of the protein. It creates one big graph that bridges the two. It draws connections between the drug's atoms and the protein's atoms based on how close they are in 3D space.
So it's mapping the social network of the entire binding site.
Exactly. It's learning the conversation that's happening between the drug and the protein. It's not just looking at the shape, it's looking at the electrostatic conversation. You know, I'm a little bit positive, you're a little bit negative, let's stick together.
And it learns this just from the data.
It learns this just from the data, and the results are really impressive. The AGIMA-Score outperformed a lot of other models in virtual screening tasks, like finding new compounds that bind to EGFR, which is a major cancer target. It's learning the nuanced physics of atomic interaction without ever being explicitly taught physics.
That's just wild. But we're not just finding needles anymore, are we? I mean, the revolution isn't just about a better search function. It's about creation. This is the big shift to generative AI.
This is the ChatGPT for molecules moment.
So instead of asking ChatGPT to write a poem about a cat in the style of Shakespeare, I'm asking an AI to write me a molecule that kills bacteria but doesn't harm the liver.
That is essentially it. We call it the novo design, which just means from the new. There are algorithms like RELEASE, that stands for reinforcement learning for structural evolution, that can literally write new chemical structures that have never existed before in nature or in any lab.
But how does it know what to write? How do you teach it what a good drug looks like?
You give it a reward function. It's just like training a dog. If the dog sits, you give it a treat. With the AI, you give it a list of objectives. You say, I want a molecule that binds really tightly to this target, is soluble in water, can pass through the cell membrane, and is not toxic.
Okay.
The AI starts by just assembling atoms randomly at first.
Then most of them are probably total garbage.
Complete and utter garbage. Unstable, toxic, useless. But every once in a while, it stumbles upon a molecule that gets a little bit closer to one of the goals, and you give it a mathematical reward. If it drifts away, you give it a penalty. And over millions and millions of cycles, it learns. It evolves toward a perfect molecule that satisfies all of your conditions simultaneously.
So I'm really moving from screening, which is looking through a library of things that already exist, to designing, writing the book from scratch.
That is the paradigm shift. And it changes the economics of discovery completely. You are no longer limited by what chemicals you happen to have on the shelf. You are limited only by the laws of chemistry itself.
But, and there's always a but, with this stuff, this brings us right back to the data problem. If the AI learns from bad data, it's going to design bad drugs.
Right, the veracity problem all over again.
And if it hallucinates a beautiful molecule that looks perfect on the screen but can't actually be made in a real-world lab, it's completely useless.
The makeability problem, yeah. Just because a computer can draw it doesn't mean a chemist can actually mix it.
So this brings us to section 4 of our discussion, data management. Now, I know data management sounds like the most boring part of the meeting.
It's not the sexiest part, no.
But based on everything we've read,
yeah,
it's the guardrail that's keeping this whole high-speed train on the tracks.
it is absolutely critical. Without good data management, all this powerful AI is just a very expensive toy.
A good example of this was the read-across method that was mentioned in the sources. This is a technique that's actually changing how regulators work.
It is, and it's a huge deal. So read across works like this. Let's say you have a brand new chemical, and you don't know if it causes liver damage. In the old days, you'd have one option.
Feed it to a lot of rats.
Exactly, which is slow, it's expensive, and it's ethically very difficult.
Right.
But with read across, you look at your massive, clean database. You find 10 other chemicals that look very, very similar to your new one, its chemical cousins. And you see that all 10 of them have been tested, and they are all safe for the liver.
So you can infer, with a high degree of confidence, that your new one is probably safe too.
Roughly, yes. It's more sophisticated than that. It looks at biological similarity as well. But that's the principle. And here's the big news for 2025 and 2026. The ECHA, the European Chemical Agency, now officially accepts read-across dossiers.
Wait, that's actually huge. That means the regulators are trusting the computer's prediction instead of demanding a new animal test.
That is a massive shift. It has the potential to dramatically reduce animal testing and speed up the regulatory process. But, and this is the big flashing red light caveat, it only works if the underlying data on the cousins is perfectly curated and correct.
If that data is wrong, your prediction is wrong.
And people could get hurt. Which brings us right back to curation, the difference between a data dumping ground and a data museum.
PubChem is the dumping ground where users can upload anything.
And Che-Bell is the museum, with its manual expert curation. But we're also seeing changes happening in the lab itself to improve data quality right at the source.
I have to mention the detail about the color-coded silicone bands. I honestly laughed when I read that. We're sitting here talking about quantum computers and generative AI, and one of the big innovations is fancy rubber bands.
Hey, don't knock the rubber bands. Look, think about the human element. It's 3:00 a.m. A postdoc has been pipetting for 6 hours straight. They're exhausted. Their handwriting gets sloppy. They mean to write a 7 on the side of a test tube, but it looks kind of like a 1.
And boom, that sample is now mislabeled.
And that single wrong data point gets fed into the database, and it lives there forever. That one tiny error could confuse an AI model for years.
So companies like Eppendorf and SPT LabTech are pushing lab automation. But sometimes the simple low-tech fix is just as important.
A color-coded band, instead of messy handwriting, protects the data. It protects the high-tech output from simple human exhaustion. And full automation, having robots do the pipetting instead of tired humans, it standardizes how the data is created in the first place, which makes the whole haystack cleaner from the start.
Okay, so we've got the big data, we have the cryo-EM eyes, and we have the AI brains. But there seems to be one final frontier in this revolution. We're hitting the wall of what normal computers can actually do.
We're hitting the classical limit. This is the quantum leap.
We have supercomputers. We have these massive cloud clusters of GPUs. Why on earth do we need quantum computers? What can they possibly do that a supercomputer can't?
Because classical computers, the ones we all use that run on zeros and ones, are fundamentally bad at simulating nature.
Why?
Because nature isn't binary, nature is quantum.
What does it actually mean, nature is quantum?
Okay, think about an electron in an atom. In a classical computer, a bit is a switch. It is either a 0 or it is a 1. It's definite. But an electron in a real atom doesn't exist in just one place. It exists in a cloud of probability. It is here and it is there at the exact same time. It's a blur of potential.
And when a drug binds to a protein, it is all about those electrons interacting with each other.
Precisely. To simulate a drug interaction perfectly, what scientists call molecular dynamics, you need to calculate the position and the energy of every single electron. And classical computers just can't do it. The math is too hard. They have to approximate or guess because they can't handle that fundamental uncertainty of nature.
It's like trying to simulate the smooth flow of water using only square Lego blocks. You can get the general shape right, but you're always going to miss the subtle fluid dynamics.
That is a fantastic analogy, yes. You miss the flow. Quantum computers use qubits, which, by their very nature, can exist in multiple states at once. They can represent that uncertainty directly. They can simulate nature as nature actually is.
So what does this mean for drug discovery in practical terms?
In practical terms, it means precision. It means we can calculate binding affinity, how sticky a drug is to its target, with perfect or near-perfect accuracy. IBM has set this public goal to speed up molecular dynamics simulations by orders of magnitude, and they are making real progress.
But is this actually happening now? Or is this still theoretical? Because quantum computing has felt like it's 5 years away for the last 20 years.
It's finally emerging now. 2025 and 2026 are being called the inflection years by many in the field. We aren't running entire drug discovery campaigns on quantum computers yet, but what we are seeing is the rise of hybrid approaches.
Hybrid.
Yes. Companies like Model Medicines and national labs like Argonne are using classical supercomputers for the heavy lifting, sorting through 1 billion molecules, and then using the early quantum computers for the really tricky final bits.
So calculating the exact electron interactions for just the top 10 best candidates.
Exactly. It's about validating the top drug targets in silico in the computer before you ever move to testing in vivo in a living body.
That's the holy grail, isn't it? If you can simulate the physics perfectly, you don't need the petri dish until the very, very end.
That's the goal.
So let's bring all of this together. We have covered a lot of ground here. Big data, cryo-EM, AI, quantum. But the real magic isn't in any single one of these tools. It's in how they all fit together.
It's the convergence. That is absolutely the word of the year.
So walk us through the workflow of the future, or really the workflow of the cutting edge today. How do all these pieces fit together to tackle a disease?
Okay. Let's imagine a patient comes in with a rare, aggressive genetic cancer.
Right.
Step 1, genomics. We sequence their tumor's DNA, and we identify a specific mutated protein that's driving the cancer. We have our target.
Step 1. Got it.
Step 2, AlphaFold 3. We plug that genetic sequence for the mutated protein into the AI, and within minutes it predicts the 3D structure of that specific mutated protein.
Okay, so now we have a picture of the enemy.
Exactly. Step 3, cryo-EM. We take a sample of the actual protein from the patient, we flash freeze it, and we use the electron microscope process via cryocloud to get the experimental structure.
To validate the AI's prediction.
To validate it, and crucially, to see how it moves. And maybe in that movie we see a hidden pocket, a vulnerability that only opens and closes for a nanosecond. A pocket AlphaFold might have missed.
Got it. A weak spot.
Step 4, generative AI. We take that structure with its newly discovered weak spot and we tell the AI, design a molecule that fits perfectly in that pocket and locks it shut. The AI hallucinates 1 million options and uses that reward function to find the absolute best one.
Okay.
Step 5. Quantum computing. We take that single best candidate and we use a quantum computer to simulate the electron interactions with perfect physical accuracy. We confirm that it binds super tight and, just as importantly, that it doesn't accidentally bind to anything else, like a crucial protein in the heart or the liver.
To predict side effects before they happen. And finally.
Step 6. Lab automation. A robot in a lab automatically synthesizes that one perfect molecule, tests it on cancer cells in a dish, and feeds that real-world data right back into the system. If it works, it moves toward trials. If it fails for some unexpected reason, the AI learns why and redesigns it instantly.
It's a closed loop, a self-improving discovery engine.
And that entire process, which used to take 5 years of painstaking trial and error, now takes a few weeks.
I have to ask the question that I'm sure is on a lot of people's minds listening to this. Does this replace the scientists? Is the human expert out of a job?
No, absolutely not. I love the quote I saw from the ELR Drug Discovery 2025 conference. Automation is the easy bit. Thinking is the hard bit. The goal of all this technology is to free the brilliant scientists from the repetitive manual labor of pipetting so they have more time to think. We need them to ask the right questions, to have the biological insights. The AI is a tool, not a replacement.
And we need what some people are calling transparent AI, right? We need to be able to look inside the box and understand why the computer picked that specific drug.
That is absolutely essential for trust. Companies are working hard on this. If an AI says, this molecule will cure cancer, a scientist needs to be able to ask why and get a clear biological answer, not just a bunch of math. Because at the end of the day, a human is going to take that drug. A human doctor is going to prescribe it. Trust is the final currency.
So as we wrap up this incredible exploration, what's the final thought? What's the big takeaway from all this?
I'll leave you with a provocative thought. We are steadily moving toward a world where a new drug is designed entirely on a computer, and it works perfectly the very first time it enters a human body. Now, we aren't there yet, not even close. We still need clinical trials. We still have massive safety hurdles. But for the first time, all the pieces of the puzzle, the eyes from cryo-EM, the brain from AI, the memory from big data, and the fundamental physics from quantum, they are all finally on the table at the same time.
We aren't just hunting for needles in haystacks anymore.
We're engineering the needles from scratch.
So whether you are prepping for a meeting, investing in this space, or you're just curious about the future of your own health, the takeaway seems pretty clear. Biology is no longer just about observation. It's about design.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support.
And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content and support us in Patreon.
Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.